Hi everyone, so our group did RNAi Knockdown. So generally the purpose of RNAi Knockdown is that RNAi stands for RNA interference. Knockdown is also called gene silencing. So this is a really big idea on the fact that knockdown is not the same thing as knocking out a gene. So it's a useful way to see which proteins are actually essential and what happens when genes are not able to go through translation, or in other words, are not able to become functional proteins. So it's a highly specific way to inhibit translation, and basically it's when two identical strands fold on top of each other to create a double-stranded RNA, which is targeted by the body to be destroyed. A super simple example of this is if you were to use RNAi to silence a particular gene in a fly embryo, and that embryo grows into an adult fly without wings. You could then maybe assume that that gene you silenced was involved with wing development.
Okay, so in order to perform RNAi knockdown, you're going to need to produce the dsRNA that corresponds to the target gene. You can produce it in a few different ways, some of which are in vivo, but I'll be talking about the in vitro method. In this case, you'd start with 2 DNA templates, each having the gene sequence that you're interested in exploring, but just in opposite orientations like shown. Then you'd mix the DNA templates with an enzyme mix containing RNA polymerase to induce the transcription of these complementary RNA strands. Then you'll mix equal volumes of your RNA reactions and heat it and then cool it in order to help anneal the RNA strands into this double-stranded RNA, which is what you want. After that, you can add some nucleases that will remove the DNA templates and then any single-stranded RNA that did not anneal together, and then purify your dsRNA by precipitating, washing, and then resuspending in nuclease-free water. And there's also a few different ways that you can administer your dsRNA to your test organisms or test cells. One way is to directly inject your dsRNA using a capillary needle. And then another way is to culture your embryos in a medium that contains the dsRNA so that they take up the dsRNA when they take up their nutrients from the medium. So once the dsRNA has been introduced into the cell, they'll be cleaved into these small inhibitory RNAs or siRNAs by a ribonuclease called dicer, and this is just an enzyme that's found in basically all eukaryotes. After that, the siRNA will complex with the RNA-induced silencing complex, also known as RISC. RISC will then just use one strand of the siRNA to hybridize with the mRNA produced by the target gene. And once that complex has been made, it will degrade the target mRNA, which will inhibit translation and inhibit the function of the target gene. So in order to assess whether or not your RNAi knockdown was successful in silencing the target gene, you can do things like reverse transcription PCR or Western blot analysis to confirm that there was a decrease in either the mRNA or protein levels associated with your target gene. And then finally, you can examine your test organism for any changes or abnormalities that might indicate what the target gene had been responsible for. We'll get more into the details of the analysis when we talk about our paper, but the big idea here is that RNAi knockdown does not alter the target gene itself, but it does alter gene expression by degrading its mRNA.
Okay, so a super simple analogy that explains RNAi would be the X you decided to block on your cell phone. So basically all the phone contacts you have are separate functional genes, and all of those genes can send text messages or be translated into functional proteins. But if you decided to block your X, then that's a good example of RNA knockdown. They can try to send you a text, but nothing's going to come through. They're still alive, they're still functioning, and the gene still exists. Their contact could still be in your phone, but it's just blocked. But if you also decided that you didn't hate your ex as much as you thought you did, then you could unblock them and they would be able to send you text again. Or in the case of RNAi knockdown, the gene could be later translated again into a functional protein at a later time.
The paper that we looked at that uses this method was called Role of adenylate kinase 1 in the integument development of Schistosoma japonicum schistosomula. This is a parasite that is an agent of zoonotic disease, and schistosomula is a state of growth and reproduction. They hypothesized that AK1 protein in Schistosomula japonicum is implicated in schistosomula development and integument growth. This hypothesis was further developed in the researchers' previous study that used immunofluorescence to show that AK1 was localized in different tissues during the first 21 days of development. In order to do this, an AK1 specific double-strand RNA and a negative control of EGFP double-strand RNA were synthesized in vitro. EGFP is a non-existent gene in S. japonicum. A blank control with no double-strand RNA was added to the final sample. These three groups were used in RNAi knockdown. Western blot analysis was performed on AK1 to measure the efficacy of gene silencing. Next, SEM and TEM images were taken in order to measure growth and integument thickness. And finally, the tunnel method was used to evaluate the rate of apoptosis after gene silencing.
Okay, so moving on to the first part of the results. So first of all, let's go over the Western blots. If you look over at figure 2, you can see that the group treated with RNAi knockdown, that's SJAK1 group, on average produced 66% less of the AK1 protein than the native control group, the EGFP. Next, the tunnel assay. So cells from the SJAK1 RNAi group had a 35% apoptotic rate, you can see that in figure 3, while cells from the native control group only had a rate of about 11%, and the cells from the general control group without any dsRNA in it at all had an even lower apoptotic rate. Next, the second part of the results. So for this, AK1 mRNA levels were measured over an 18-day period of schistosomula development, ending at day 21. It was determined that AK1 mRNA transcript levels were highest at day 21, as you can see in figure 4. And on to part 3 of the results, our last part of the results. So these results both had to do with the integument development of the schistosomulas. So first, immunolocalization assays indicate that AK1 proteins were mostly concentrated in the integuments of the schistosomula, and also had their highest levels of protein concentration during day 21 of growth. Finally, electron microscopy of SJAK1 RNAi knockdown group showed that the integuments were ruptured or in some other way improperly developing. You can see that in figure 5. So now that we have these results, what conclusions can be drawn for them? So there are two major conclusions which the researchers came to. The first was that the AK1 gene of Schistosoma japonicum was successfully knocked down. This was determined from the Western blot results. Remember that 66% decrease in AK1 protein production in the RNAi treated group. The second conclusion is that SJAK1 is a key gene in Schistosoma japonicum schistosomula growth, specifically in the proper development of the integument. This was supported by the fact that SJAK1 mRNA transcripts were found in all stages of the schistosomula development. The integuments of the RNAi treated groups did not develop properly as seen by electron microscopy, and that the cells from the SJAK1 RNAi treated group underwent apoptosis with a greater frequency than either of the control groups.